Well, good morning. Great Salt Lake, who lives here? Well, what we're going to talk about is why do we even want to know, right? And I would say why we need to know. And then kind of tip in my hand, then I'm going to talk about the Aquatic Life Use Workshop for Great Salt Lake that was held in 2015. I'll tell you about what we learned at that workshop. And then I'll tell you where we're headed from here and why. This goes to the why do we need to know. On the right shows you how many standards, water quality standards we have for Great Salt Lake. We have one. For all the rest of Utah's water, we have about 170 pollutants identified, and we have numbers that say this is the maximum you can have in the water, but only one for Great Salt Lake. Why is that? Well, oh, did I hit the scary button? Go back to water quality standards and what they are. When most people think, so there's three elements to them: designated uses, criteria, and the anti-degradation policy. And since I can't say that word, and I'm not going to talk about that, that's off the table. What most people think of when you think water quality standards, they think of this, and specifically numeric criteria. This is the concentration that we say is allowable in the water. There's also narrative criteria that says you can't do anything bad to the water. But these are tied directly back to the designated uses. So we need to know what our designated uses are before we can develop criteria to protect it. Designated uses are our management objectives for the water. And these protection and propagation of aquatic life and recreation, that's our so-called fishable, swimmable uses that were required by federal law to have, but we also have some additional ones. We have one for water that's going to be used for drinking water, and we have one for agricultural use of our water for both irrigation and stock water. So if we know what this is, for instance, drinking water, we would say arsenic can't be any higher than 10 micrograms per liter or 10 ppb. That would be our criteria for drinking water. If it's to protect the aquatic life, the fish and bugs in there, well then we say it can be 150 parts per billion or micrograms per liter. So I just want you to realize we need to define these uses so we know what we're trying to protect, then we can figure out how to protect it. So I go to our water quality standards and say, have we designated beneficial uses for Great Salt Lake? And yes, we have. So for Gilbert Bay, it's protected for waterfowl, shorebirds, and other water-oriented wildlife, including their necessary food chain. And Gilbert Bay is this area right here, and that's the one we were mainly talking about in the last talk with the brine shrimp and the brine flies. And then I look at Gunnison Bay, and the standards say protected for waterfowl, shorebirds, and other water-oriented wildlife. And that's up here. And gee, Bear River Bay says the same thing, as does Farmington Bay and the transitional waters, which are the mud flats between the open water. So all these have the same uses designated for them, but we know they're not all the same. This is our main brine shrimp place down here. Gunnison Bay is very salty, mainly archaea, a bacteria-like organism, the pink stuff. And Bear River Bay is pretty fresh. And Farmington Bay kind of gradates from fresh at the south end, and it gets saltier as it gets up towards the Antelope Island Causeway and comes under the influence of Gilbert Bay. So these are all very different ecosystems, yet we have the same descriptions in there. So what we really want to know then is, when we say this, who do we mean? Now, waterfowl and shorebirds, they're pretty mobile. They'll go where the food is, so they go all over the lake. So not particularly concerned about them. The GSLEP program that we heard about, they count birds. There's several people out there keeping their eyes on birds, and we have them pretty well characterized. What we don't know is this necessary food chain stuff as well. We know brine shrimp and brine flies here, not so much here. So we decided to do research, and we went through the literature, and just like we heard Dr. Bolovsky in the last talk, there's not a lot out there. Surprising, but there's just not a lot there. And when we did this, we also realized there's a lot of information out there that we don't have. So a lot of the people that have presented here, we know there's research going on in the lake, but a lot of that doesn't make it into the primary literature, and a lot of it's in people's heads. So we thought, why don't we have all these people come in and tell us what they know? We didn't even have to threaten them. So in March of 2015, we hosted an aquatic life use workshop for Great Salt Lake. We invited everybody that does research on the lake. We had already compiled what the primary literature and secondary literature we could find had, and we said, let's talk about, let's try to define. So our objectives were to develop the resident species list of aquatic organisms for the open waters of Farmington Bay, Bear River Bay, and Gilbert Bay. We didn't specifically include Gunnison Bay because it's really unusual because of that high salinity. And the primary organisms, we're not sure how we're going to protect it. But we do know, we heard the brine shrimp, that the birds depend on them, a $600 million industry depends on them. We need to make sure that we're adequately protecting the lake. I'm going to talk a little bit more about resident species in the next slide, so keep that in mind. But we also asked people for salinity tolerances and life cycle information. Our working hypothesis is that the lake ecosystem is primarily governed by salinity. So why it's different in Gilbert Bay and Bear River Bay can be explained by the differences in salinity. We had a good turnout, 40 people came. We did a final report. In that report, we tried to document all the conversations we had, the oral information people provided, and then we also asked for any databases they had, and we compiled all those into an electronic database that is accessible to anybody that's interested in doing research. And we're hoping to keep this will be a living database, and we just keep adding as we go. So residents. EPA defines residents as species that are usually present at the site. That's pretty easy. Are present at the site only seasonally due to migration. This would be our birds, for instance. Some of them only migrate here. They don't live here year round. You also see this with things like salmon that come through, or their young stage is freshwater and then they migrate to saltwater. But for aquatic life, we don't have a lot in that category for Great Salt Lake, at least that we're aware of yet. They can be present at the site intermittently because they periodically return or extend their ranges. I think we have this a lot at Great Salt Lake because the lake varies over time and space with salinity, that when things are good, these organisms come back, these ones move out, and that happens all the time at the lake. So the problem is, though, when we go out there and just take a snapshot in time, we think, oh, that's what lives here. But tomorrow that could be different. The other ones they look at were present in the site, not currently present due to degraded conditions, expected to return, and are present in nearby waters, but they're not at the site because of degraded conditions. We're unaware of any situation that we have this, but part of that is because we don't have good records and we don't know what used to be. So we really don't know what's supposed to be there. For a freshwater stream, we can tell you pretty much, oh yeah, that type of stream should have this type. We can't do that with Great Salt Lake. We don't have reference sites. There's other saline lakes around the world, but they're not like Great Salt Lake. They're all unique. So that's a problem for us to, we have to just start today and base what we're saying should be there on what is there. So talk a little bit about workshop results, and since some of you probably have been out of high school for a little while, taxonomy just real quick. It goes kingdom, right, plants or animals, and phylum, class, order, family, genus, species. What we're mostly want to know is down here, family down to species. And the reason is because using EPA methods, if you have a species and there is a toxicity value, a toxicity test result for a different species, but it's in the same family, you then use that other one as a surrogate because you have data for it. There's a lot of controversy over that, but that's been the way it's been since 1984. So we care really at family. What did we find? There's 7 families of fish in Great Salt Lake. That surprises a lot of people. We have 54 families of invertebrates, 23 families of phytoplankton. And that I already told you we have compiled, and if you're interested, you can go to the website here and see all this, the report and the database. So looking at it on a individual bay, you can see at the family level, Gilbert Bay is the least diverse. And then it jumps up, Bear River is the highest, and then Farmington Bay. That same pattern is repeated again when we go down to genera. The other interesting thing about this graph is it shows that there's not that many more genera than families. So there's only a few representatives of that family in the lake. A lot of times you'll have a whole, you could have 50 species, especially if we're talking something like chironomids or like that. There's a whole bunch of species to one family. Well, we don't really see that in the lake. Generally, it's only a few members from each family can tolerate the conditions. What did we find out in Gilbert Bay? No fish. Although nobody's ever really looked, as in a detailed fish study, they're out there with nets, things like that. Plus the salinity is so high, you just don't expect fish. So we're concluding there are no fish. The brine fly was unique to Gilbert Bay. And we know that up in the North Arm in Gunnison Bay, there used to be brine flies because the biostromes or microbial lights that they are still there, but the brine flies are. Really nice pictures by Dr. Wurzbaugh. He's retired from Utah State now, the brine flies on the surface film. But we identified some data gaps too, and this will help us then, what do we need to know to go forward? We don't know a lot about brine flies. They're extremely, other than they're really important to birds, really, really, really important to birds. So there's something we really want to pay attention to protecting, but there's just been very little research done on brine flies. The benthic or bottom communities are not characterized well. People didn't go out and actually dredge sediment and sieve it and see what kind of bugs we have. We don't know. And then microplankton is probably underrepresented in our samples because of the nets. They're trying to catch brine shrimp. Right? We saw in the GSLEP program, the ecosystem program, they're out there towing for brine shrimp. So their mesh is probably too big to capture these little guys. In Bear River Bay, and I really like this idealized photograph of Bear River Bay or figure, and I'll show you why it's idealized in a minute. But there's Great Salt Lake minerals. This, from here back, we call Willard Spur, and that was the subject. It's right next to Willard Bay right there, and then there's the refuge. That was the subject of some intense research. Jeff Dumblaker has presented here at this conference on it. Had a lot of work there, but not much out here in what we call Bear River Bay proper. But it's the only place where we have any fish studies. And that's where a lot of these, the fish numbers come from. It's the only fish study we have for the whole lake is in this. And this one really isn't representative of the lake. It's very low salinity. So it's pretty fresh water. Plus you have some escapees from Gilbert Bay in there as well. But that's the only place. We've also found a lot of unique taxa in the invertebrates there. But again, that could be a function that it's that diverse, or simply that that's where we spend a lot of time actually trying to see who lives there year round. So, as I said, there's no fish studies for Bear River Bay proper. This is a Google Earth map. This is the refuge. These are their dikes that they have. They control water. This is that nice big blue thing that was idealized in the last one. It's pretty dry. It's really dry. There's no water there. So there's no water there. How am I going to do fish studies or look for benthic invertebrates? That's going to be a case where they would come in if conditions improve, but I won't know who comes in until conditions improve. So kind of a catch-22 there without water. And it's hard to protect water quality when there's no water to protect. So that's a dilemma. In Farmington Bay, I found more zooplankton than the other bays. And what you're looking at there, this is a NASA sent this out, probably a month ago. It's 2011 and 2016, just going back and forth. It shows you in 5 years the extent of the dewatering or, receding water in Farmington Bay. Same problem there. We still have water coming through where the Jordan comes out, but much, much less extent. There's several taxa listed here that show that were only found in Farmington Bay. And Farmington Bay receives a fair amount of research as well. Some interesting ones are that you have like the Daphnia. And one of the things we struggle with for our permits is they have to do whole effluent toxicity testing. And you want an organism that represents the water where they're going in. And that can be a problem, but this is showing that some of our standard test organisms like a Daphnia actually are present there. So the lake as a whole, what did we identify? Well, I said we got the benthic invertebrates, including the biostrom communities or microbialites. There's a lot of disagreement on what they should be called. They're from precipitation from the blue-green algae on them. That's a cyanobacteria. In this case, it's not one of the toxic ones, but it's one the brine flies need to eat. And this is a picture of those. You can go out to the lake and see these in quite a few places. Jeff Dunbaker was just telling me yesterday he was out and found, and these were, the ones he saw were probably 4 to 5 feet high, these cones coming up out of the water. So we don't know them yet. We have to work on them. Microplankton. This is a picture of microplankton, but they're too small for you to see. But you'll have to trust me. So we just need better nets for those, finer nets. We also need to characterize the fish communities in Bear River and Farmington Bay. Now these out-of-staters here, they think there's fish in Great Salt Lake. But what do they know? They're from out of state. We do see pelicans actively feeding out there. We know there's fish in there. People see the fish, but we don't know how many, what kind, because nobody's ever looked. And so that's a data gap we have to work through. And then also the salinity tolerances of these. Where is it going to change? When the salinity changes, where is it going to change to another system? The first problem we have is everybody measures salinity different. You think it would be a really easy, straightforward measurement. It's not. And experts are still arguing about the best salinity measure. So we did, in our report, we did get the conversions. There's ways to convert back and forth. They're imperfect, but that way we can at least use the data everybody collects. The other problem we have is you really need experimental studies to figure out salinity tolerances. So you put some bugs in there, you increase the salinity, oh, they're still doing okay, and increase it. Basically a toxicity test for salinity. You find out where their threshold is, where they will cease to grow, reproduce. But we need field studies to put that into ecological context. And some of the best are mesocosms where we incorporate it. And just to give you an example, one of the things that came out in our workshop was Dr. Belovsky pointed out, yeah, brine shrimp will live in that fresh water just fine. They're happy as could be. The problem is the carixids can live there now. And so when the carixids are there, they basically eat all the brine shrimp. So while the brine shrimp can survive, it's probably not relevant for the lake because the carixids will predate on them. So where are we going? Well, obviously we got some more work to do, and we're hoping that this will help focus future research effort. We're going to take what we do know about Farmington Bay and Bear River Bay and see how the families, the taxonomic families we have here compared to the taxonomic families that EPA's used to derive freshwater numeric criteria, and see how close we are. We'll probably identify more data gaps. A big one is develop implementation methods to address temporal and spatial variation. If you're a discharger, most of them will meet whatever standards you tell us we have to meet, but we can't have you changing that every year because the lake changed, the salinity changed. So we have to figure out a way to apply these in a way that makes sense but still protects the water quality. We anticipate for Bear River and Farmington Bay, we can use EPA recalculation procedures, which is simply, they have a big toxicity base. They base these national numeric criteria on. We just take out everything we don't have in there for our species, our families, and then recalculate the criteria and move right along. For Gilbert Bay, it's a special case. We have to do actual toxicity testing. And we're engaged in those right now with brine shrimp and brine flies. Eventually we'll have to do algae. We'll actually measure how much arsenic to hurt them, how much copper, because that's not information we can get anywhere else. And hopefully someday the lake will look like this again, full up. That would be a better situation. So that's all I had. I want to thank all the workshop participants listed here. Without them, this never could have happened. Questions? Wow, you guys are easy.
Chris, who's going to do the toxicity testing in Gilbert Bay?
Right now, Gary Belovsky did the organism rearing, and so he cultured the brine shrimp. Well, they're easy. The brine flies in his lab, and we're actually right now selecting it. It'll be either a commercial or a university laboratory to do it. It's more research because it's method development. We can't just take a test off the shelf because EPA, well, they specifically forbid using brine shrimp because they're relatively insensitive, which is a good thing for us. It means they're probably not, but nobody's ever done a brine fly. So it's a lot of method development as well. And we've had plenty of trials and tribulations associated with that. One of them is just making artificial Great Salt Lake water. We can't get the salt to dissolve in it at 13, 12% salinity, and we have 15, 18, 19 out there, 27 in Gilbert Bay. that much salt will go into solution. So we're missing something, right? And it could be dissolved organic carbon. The funding comes from the Water Quality Board to get us started. So they gave us a grant to pursue this. This is all part in the Great Salt Lake Water Quality Strategy. We have this mapped out what we want to do, and a lot of it's dependent on funding availability. And hopefully one of the things that will come out of it is a specific whole effluent toxicity test organism for Gilbert Bay discharges. That's the hope. But I don't know if we'll still be here. I'm not going to call you on you till he gets there with the microphone.
Just real quickly, AB, you did the 3 bays. Are you going to extend this to eventually to the fringe? This kind of study?
You know, the fringe is a little more challenging in that we have the same fringe specified for the whole lake. So the fringe in Gilbert Bay is very different than the fringe in Farmington Bay, where we have the beautiful wetlands and shorebirds everywhere. And, you know, Gilbert Bay is a little, I hate to use the word sterile, but it's not. So we've just been looking at those on a case-by-case basis. And some of those are effluent because they, for instance, Kennecott used to discharge right into Gilbert Bay. I mean, the pipe came out, it went right in the water. It's 1.5 miles, 2 miles out to the open water now. So there's a channel and a stream. We're going to try to walk before we run.
All right. Thank you, Chris. We're out of time.